Introduction to the Physiology Part 3. As we have gone through with the second part up to the secondary membrane transport system, secondary membrane transport system. And now today we'll start with the membrane action potential, all of those which is rest in the introduction to the physiology. What is the membrane transport? Who can say what is the membrane transport system?
Sir,
What is the
membrane transport is the process through which substances travel.
through which substance?
Travel.
Sorry, I didn't get it.
Sir, membrane transport is the process through which substances travel.
Substances travel uh, across which travels across which
Across
across
membranes.
the across the membranes or across the cells. Okay, membranes of the cells. So they process the cells. So membrane transport is the process. As I talked to you, say like this: membrane transport in the process is the process by which different substances are transported across the biological membrane in a biological system.
Yes,
Okay.
sir. Yes, sir.
What are the types of membrane transport? You can say. What are the types of membrane transport?
Sir, two types of membrane transport are there. Sir, passive and active. Sir, passive transport, simple diffusion, osmosis, and facilitated transport. And sir, active transport, there are two types, pump and vesicle. Pump, primary and secondary, and vesicle, exocytosis and endocytosis. Sir, exocytosis, neurotransmitter, hormones, enzymes, and protein. And endocytosis, pinocytes, phagocytes, receptor mediated.
Yes, but in active transport, what are the types of active transport?
Sir, pump and vesicle.
What are the types of active transport?
Sir, pump and vesicle.
Say
Primary
like this:
active.
primary active transport and secondary active transport system. Okay.
Okay, sir.
Primary active transport, the pump system like sodium, potassium pump, calcium pump, hydrogen pump, all of those in primary active transport. Okay. And secondary active
Yes, sir.
transport, there is a co-transport and counter-transport. Co-transport is sodium glucose co-transport, sodium amino acid co-transport, and counter-transport is sodium calcium counter-transport, sodium hydrogen counter-transport. Co-transport means it goes in the same way. The same way, the both gets entry in the same way by the secondary active transport, and counter-transport is the uh, is going in a counter way, like the sodium in and potassium out, sodium in, hydrogen out. This is the counter transport. Okay.
Yes, sir.
Other transport processes are, as you said, exocytosis and endocytosis. Exocytosis, through exocytosis, what gets transport? It's
Sir,
transport.
neurotransmitter,
Yes.
hormones, enzymes, and proteins.
Yes, hormones, enzymes, and proteins are released from the cells by this process. And endocytosis has two process. Endocytosis has two process. What
Pinocytes,
are those? Yes,
pinocytosis, and
yes.
phagocytosis.
Pinocytosis and phagocytosis. What are the other name of pinocytosis? You can say.
Pinocytosis, cell drinking, and phagocytosis, cell.
Yes. Because why it is called cell eating phagocytosis?
Sir, phagocyte because it ingests bacteria, dead cells, and tissues.
Exactly. Do you know what is transcytosis? Who knows
No,
what is
sir.
transcytosis? Anyone? Transcytosis is a type of transcellular transport in which various macromolecules are transported across the interior of the cell. Macromolecules are captured in vesicles in the cells on the sides of the cell, drawn across the cell and ejected on other sides. For example, examples of macromolecules may be include immunoglobulin A, immunoglobulin. That means a immunomediator, immunoglobulin A, transferrin, and insulin. Yes, the insulin, transferrin, and immunoglobulin A. All those are the examples of macromolecules. Those macromolecules are processed by, as captured as vesicles and transported to one cell to another, especially in the interior of the cell. This is called transcytosis. Am I clear?
Yes, sir.
In Viva, you may be asked how are the water soluble substances transported through the cell membrane? What will be the answer? Water soluble substances transported through the cell membrane. How?
Sir, simple diffusion,
Yes, by
uh, protein channel.
through protein channel, and by facilitated diffusion by
Carrier
through
protein.
Carrier
carrier,
protein.
and active transport through,
Carrier protein.
yes, carrier protein also. So how lipid soluble substances are transported? By how lipid soluble substances are transported?
By diffusion.
Hello?
By
Yes, sir.
diffusion.
This ingestion of the large particles such as bacteria, whole cells, or portions of the degenerating tissue. So this is the phagocytosis. That means phagocytosis is done to the large particles like bacteria, whole cells, or even portion of the degenerating tissue. Degenerating tissue. Do you know what is degenerating tissue? Can you see the slide clearly?
Yes, sir.
Can you do you know portions of the degenerating tissue? The degenerating tissue. What do you mean by degenerating tissue? Can you anyone?
Sir,
Whereby?
breaking cells which are breaking down or which are dying.
Maybe yes. You could say it. Degenerating cells are necrotic tissue or the cells, yes, the tissues which are dying or may injured. Okay, so these cells, or in case of foreign body, also are attacked by the phagocytes, and phagocytosis occurs. Okay, so that our body may cure itself by this pinocytosis and phagocytosis. Importance of endocytosis is the normal in the normal consumption. What is the importance of endocytosis? Mainly, pinocytosis is the only means by which most large macromolecules, such as most protein molecules, can enter the cell. Number 1 point. And number 2 point is microbial agents and foreign particles are destroyed by the phagocytes. And what is exocytosis? Very large particles like exit the cells by a specialized function of the cells, as it is endocytosis and exocytosis. The question will be illustrate the process of endocytosis and exocytosis. Endocytosis has two brothers, pinocytosis and phagocytosis, and exocytosis itself it takes the very large particles exit the cell by a specialized function of the cell membrane called exocytosis. The importance is neurotransmitter, hormones, enzymes, proteins are released from the cells by this. Am I clear up to this?
Yes, sir.
So, this is named the energy dependent transport process across the cell. Energy dependent transport process are the simple diffusion, facilitated diffusion, and active transport. What are the amongst them? What is the energy dependent one? Okay. What are the energy dependent one?
Active transport.
Yes. So you can see in the picture, energy dependent is the active transport system. You will be asked in the Viva define carrier protein and mention its type with example. We'll say carrier protein. The who can say what is carrier protein? Carrier protein. Okay, note down. Carrier protein. Integral protein act as carrier protein. Okay, for transporting substance. That you know, we have two types of protein: integral protein, and another one is what? Peripheral protein, isn't it? So,
Yes, sir.
Yes, sir.
so integral proteins act as a carrier protein for transporting this substance. Not peripheral protein, integral protein act as carrier protein for transporting substance. In this carrier protein has a two types, uniport and symport. The carrier protein transporting only one substance is called uniport. For example, calcium pump. It only transports the calcium. So calcium pump is uniport carrier protein. And symport, the carrier protein transporting more than one substance is called symport. And for example, sodium iodine transport symporter, sodium iodine symporter, and sodium glucose co-transport. Another one is antiport. These are the transport proteins that exchange one substance for another. The sodium-potassium pump is a typical antiport. That means one substance goes out and another substance goes in. It moves like in sodium-potassium pump. What occurs? It moves free sodium out of the cells in exchange for two potassium. That it moves into the cell, isn't it?
Yes,
Am I
sir.
clear?
Yes, sir.
For Viva, you may ask in the, as another question. Okay. So the short in case of short note, you may get pinocytosis in short note. Pinocytosis is the cell drinking injection of the substance not visible under microscope. Mechanism is what? In this process, the substance like protein, for example, attaches to the brush border of the luminal membrane, attaches to the brush border, that means the membrane. So luminal membrane, and this portion of membrane then invaginates to the interior of the cells until it is completely pinched off and a vesicle is formed containing the substance like protein. Because pinocytosis requires energy and it is considered a form of active transport. So pinocytosis is a considered as a active transport system. And you will be asked that what is diffusion? Do you know what is diffusion? Show the next slide, please, Ajim.
Yes, sir.
Show the next slide. What is diffusion?
Sir, the net movement of particles from an area of higher concentration to an area of lower concentration.
Yes. So the diffusion may be regarded as the movement of the solute molecules by the normal kinetic motion from a higher concentration to the lower concentration. Types, what may be the types of diffusion? What are the types of diffusion?
Simple or facilitated.
Simple facility.
Simple diffusion and facilitated diffusion. Ajim, show the next slide, please. This is gone. Uniport, symport, antiport have completed. Yes. So simple diffusion and facilitated diffusion. What are the simple diffusion? It doesn't require any carrier protein. So it doesn't require any carrier protein mainly. Diffusion, simple diffusion of oxygen, carbon dioxide, etc. are simple diffusion for example. And facilitated diffusion, what are the facilitated diffusion requires carrier protein? Facilitated diffusion of the glucose is the it is transported by a carrier protein, what is called GLUT. You have GLUT1, GLUT2, GLUT3, GLUT4. All are the carrier protein which helps the glucose to get inside. So requires carrier protein. So glucose is transported to the cells by facilitated diffusion. Factors affecting the rate of diffusion. This is also important. Concentration of the difference, concentration difference. Mainly, if you have particles concentration difference, it may be if you have more concentration, it will get the rate of diffusion is proportional to the concentration difference mainly. So when there is a gradient difference, if there is a higher concentration more and lower concentration less, the gradient difference is more, the rate of diffusion will be more. If the difference gradient difference is less, the diffusion will be less. So the concentration difference is also important. And cross sectional area, the rate of diffusion is proportional to the cross sectional area. How many areas do they have, they collapse to how many area they are, they act as junction. So these are the rate of diffusion. And distance between the two regions, like a rate of diffusion is proportional, is disproportional to the distance of between the regions, distance between the regions. So if the rate if the distance is more, the rate of diffusion will be less. So these are the inversely proportional to the distance between the regions. And temperature, if the temperature is increasing, then kinetic energy also increases and increases the rate of diffusion. And
Pressure too,
size of
sir.
the particle. Sorry,
Pressure.
say it again. What?
Pressure.
I didn't get it. Moinul.
Sir, pressure. Pressure.
Noimul. If you explain rate of diffusion as J, alphabet Z, so Z is equal to minus dA into del C by del X. D is here the diffusion coefficient. So, this is not needed. Forget it. Importance of diffusion. This is important. Importance of diffusion. Exchange of oxygen and carbon dioxide in the lungs and tissues occurs by the diffusion. Absorption of pentose, mineral, water soluble vitamins, etc. from GIT, gastrointestinal tract, occurs by the diffusion. If you have any confusion, you may ask instantly. Admixture of foodstuffs, admixture of foodstuffs which with digestive juice like bile, and digestive juice like enzymes, and juice like pancreatic enzymes. All of those needs to digest your food. So admixture of the foodstuffs with the digestive system is also important for also important as they They do it by diffusion process. Exchange in capillary bed. Diffusion is also important for exchange in capillary bed. That means capillary is our in our circulatory system. The capillaries plays an important role. So capillaries exchange their substances in the blood and outside the blood by this diffusion system. So secretion of the waste products like ammonia, as you know, it is the waste products into the renal tubule occurs by diffusion. So diffusion is a very important thing to excrete our ammonias or waste products through urine. So to production of urine is mostly depends on diffusion. You will get it in the renal part in details, renal system. Diffusion of the sodium and potassium through the sodium potassium leak channel creates resting membrane potential, which you will get in details next. And in pneumonia, in case of disease condition like pneumonia, um, it increases thickness of alveolar membrane, and the alveolar membrane thickness increases. Do you know where is the alveolar membrane?
Lungs.
Who can say what is hypoxia?
Sir, lack of oxygen.
Lack of oxygen in blood or in body. Okay. So this is the importance of diffusion. And you may ask how diffusion occurs through a cell membrane. Diffusion through the cell membrane is divided into two subtypes as simple diffusion, facilitated diffusion. You will say just simple diffusion, facilitated diffusion. That's all. And simple diffusion, if you ask to explain, you will say simple diffusion means the kinetic movement of the molecules ions occurs through the membrane opening, through a membrane opening or through intramolecular spaces without any interaction with the carrier proteins in the membrane. The rate of diffusion is determined by the amount of substance available, the velocity of the kinetic motion, and the number and sizes of the openings in the membrane through which the molecules or ions can move. Simple diffusion can occur through the cell membrane by two pathways: through the interstitial pathway of the lipid bilayer, and if the diffusing substance is lipid soluble, only then this simple diffusion occurs, and through watery channels that penetrate all the way through some of the large transport proteins. So this is the simple diffusion. And facilitated diffusion requires interaction of the carrier protein, as I said previously. The carrier protein aids passes of molecules or ions through the membrane by binding chemically with them and shuttling them through the membrane in this form. So you need to learn about the difference between the simple diffusion and facilitated diffusion. Carrier protein is not required in case of simple diffusion, and facilitated diffusion carrier protein is required. And rate of limiting process, uh, as carrier protein is not required, so there is no rate limiting process in case of simple diffusion. And here rate limiting process is important as carrier protein is required in case of facilitated diffusion. Saturation of the receptor or carrier protein, it is saturable, simple diffusion as there is no receptor, and facilitated diffusion it is saturable as there is receptor. Rate of diffusion, the rate of diffusion is increases proportionately with the increase of substance concentration. And facility, in case of facilitated diffusion, the rate of diffusion approaches a maximum, that means velocity maximum V max, as the concentration of the diffusing substance increases. Then the rate will not further increase. Do you know what is osmosis? What is osmosis?
Sir, the diffusion of solvent molecules across a semi-permeable membrane from the region of lower solute concentration to the region of higher solute concentration.
Yes. So This osmosis occurs from the lower solute concentration to the higher solute concentration through a semi-permeable membrane. As I said in the previous class, the migration of the solvent molecules from the region of the lower solute concentration to the region of higher solute concentration across a semi-permeable membrane is called osmosis. Condition of the osmosis. What are the condition of osmosis? You must need what?
Semi-permeable membrane.
Semi-permeable membrane and a different solution of different
Different
osmotic
concentration.
pressure. Okay, different osmotic pressure is needed and different solution is also needed. So, are you changing the slides? Ajim, I'm not seeing the slides. Okay. Change the slide, please. Change this one also. Next one, please. Next one, please. So, importance of osmosis. What are the importance of osmosis? Physiological importance are absorption of the gastrointestinal tract, that means GIT. Absorption of the gastrointestinal. Do you know what are the path? What are the boundary of GIT? From which part to which part? GIT, gastrointestinal tract. Do you know what is gastrointestinal tract? Anyone? What is gastrointestinal tract?
Elementary canal.
Yes, alimentary canal. From where to where? It is called an alimentary canal.
Mouth till rectum or mouth to anus.
Yes, from oral orifice to anus. Okay, mouth to anus is the GI tract. So absorption from the GI tract. Any imbalance in osmosis or osmotic pressure in your body, you may suffer for osmotic diarrhea. So diarrhea has another name, that is osmotic diarrhea. So absorption is, if your absorption is hampered, you may suffer for osmotic diarrhea. So absorption from the GI tract is important as an importance of osmosis. So fluid exchange in the capillary bed, regulation of the urine formation, reabsorption of the CSF. Do you know what is CSF? Cerebro
Spinal fluid.
cerebrospinal fluid. Yes, CSF. So reabsorption of CSF. CSF is always produced in our body, and again it's reabsorbed. Some are excreted through our urine, and some are reabsorbed again to our body or to our brain. So this is the reabsorption of the CSF. So reabsorption is occurs by also by osmosis. Maintenance of blood pressure. Your maintenance of blood pressure also depends on osmosis. Regulation of water balance in the body. As I was saying, that diarrhea may occur due to the osmotic imbalance. So osmotic imbalance also causes the water balance difference in the body, because you cannot absorb your waters and water eating and waters also. So it is all going out through your mouth to anus. Nothing is absorbed by your body. So in case of diarrhea, osmotic diarrhea, this is happens. So physiological importance of osmosis is important. Define osmotic pressure. You will ask what is osmotic pressure. The amount of pressure required to prevent the osmosis. Amount of pressure required to prevent the osmosis is the osmotic pressure. Importance of plasma colloidal osmotic pressure. You need plasma colloidal osmotic pressure. Why? In the body, osmotic pressure is excreted by both colloids and crystalloid solutions, but only the colloidal osmotic pressure, not the crystalloid one, plays important role in maintenance of fluid volume. So fluid volume is maintained by only colloidal solution. So colloids in different body compartments control the fluid volume of those compartments. For example, plasma protein exerts osmotic pressure, which retains plasma in the blood vessels and also causes diffusion of the interstitial fluid.
from the tissues into the capillaries. Capillaries are the small blood vessels. So interstitial fluid from the tissues into the capillaries and thus maintains the plasma volume. On the other hand, intercellular proteins which control the intercellular volume. Am I clear?
Yes, sir.
Okay. So you must have to read the difference between the osmosis and diffusion. Osmosis and diffusion you will go through. And let's go to the active transport system. As we have discussed already, active transport, primary transport and secondary transport. We'll move on. We have already discussed sodium potassium pump. We'll move on to this. Show the next slide, please. And the transport process which pumps 3 sodium from the inside to outside, and at the same time 2 potassium outside to inside across the cell membrane against their concentration gradient with the active expenditure of energy by the help of the carrier protein is called sodium potassium pump. We have already discussed this pump, so just go through it, and in Viva you will be asked what is the coupling ratio of sodium potassium pump. The coupling ratio of sodium potassium pump is 3:2. That means 3 sodium out to potassium in. Importance of sodium potassium pump, physiological importance, responsible for the generation of the membrane potential, control cell volume, basis of nerve and muscle function, as I said in the previous class, so I'm not going through this. I also discussed importance of sodium potassium pump in controlling cell volume already. So show the next slide, please. Difference between the active transport and facilitated diffusion you must go through. Very important. Active transport and facilitated diffusion. Direction of the transport is usually unidirectional, and facilitated diffusion can operate bidirectionally. Movement occurs always against an electrical or chemical gradient, concentration gradient. Facilitated diffusion towards electrical or chemical gradient. Energy requirements requires energy in case of active transport, and facilitated diffusion energy is not required. Role of concentration gradient doesn't depend on these factors, active transport, because energy carrier is used directly. In case of facilitated diffusion, depends upon these factors like hydrostatic pressure and osmotic pressure. Yeah, we have gone through the secondary transport system also. Discuss the process of glucose movement across the cell membrane with diagram. This is the question came in Russia University, not in Dhaka University. Process of glucose movement across the cell membrane is a sodium glucose co-transport, facilitated diffusion, and insulin dependent transport. This occurs in the brush border of the intestinal epithelium and luminal part of the renal tubular cells and cell membrane through the sodium dependent glucose transporter. Facilitated of sodium glucose co-transport. The glucose is transported here by 3 systems: sodium glucose co-transport, another is facilitated diffusion, another is insulin dependent transport. Isn't it? The sodium glucose transport is transported by the sodium dependent glucose transporter, which helps to transport it, and facilitated diffusion glucose transporter, GLUT. We have GLUT 1, 2, 3, 4. GLUT 1, 2, 3, 4. This GLUT helps in facilitated diffusion and insulin dependent transport. Occurs in muscles and most of the cells of the body with few exception. Okay.
Yes, sir.
Show the next slide, please. Yes, this one. The membrane potential. The electrical potential difference across the membrane is called membrane potential. Transmembrane membrane potential difference or transmembrane potential gradient, you may say. The types is resting membrane potential and another is action potential. As it is saying resting membrane potential, that is resting condition, and action potential is in action or in dynamic condition. Give the importance of membrane potential. Importance of membrane potential is conduction of nerve impulse, number 1. Muscle contraction and activity of the cells such as granular cells, macrophages, ciliated cells. These are the importance of membrane potential. What is resting membrane potential? Go to the next slide, please. What is resting membrane potential? Yes, this slide. Keep this slide. Resting membrane potential is electrical potential difference across the cell membrane between the inside and outside of the cell under resting condition. So it is called resting membrane potential. Amount of resting membrane potential is large nerve fiber. In large nerve fiber is minus 90 millivolts. Always it is negative. Always it is negative. It maintains its negativity inside the cell. As in the cell, there is always negative ions. There is always lack of positive ion. So this is always negative inside the cell. So inside the cell, it maintains its resting membrane potential, which is negative. In case of large nerve fibers, it is minus 90 millivolts, and neurons minus 70 millivolts, and SA node that of the cardiac muscle, that means sinoatrial node of cardiac muscle, which initiates the heart pump ability, heart pump signals, which is the minus 55 to minus 60 millivolts. And cardiac muscle fibers and skeletal muscle fibers are 85 to minus 85 to minus 90 millivolts. Smooth muscle fibers are minus 50 millivolts. Why resting membrane potential is negative? As I said, selective permeability of the cells and sodium potassium pump. Selective permeability of the cells. The potassium ions are really very, very important question. This is also for Viva and written. Potassium ions are freely permeable, but the permeability of the sodium ions is much less. So the cell membrane is impermeable to the proteins and phosphate, and inside the cell there is increased concentration of the protein and phosphate. As protein and phosphate are negative, so these factors causes negativity inside the cells by the Gibbs-Donalds membrane equilibrium. This is called Gibbs-Donalds membrane equilibrium because inside the cell, as potassium ions are freely permeable, but the permeability of the sodium ions is much less. The cell membrane is impermeable to the proteins and phosphates, and inside the cell there is increased concentration of the protein and phosphate. These factors causes negativity inside the cell. And sodium potassium pump, during each time 3 sodium goes out and 2 potassium goes in, so that's creating a negativity inside. So that's how they maintain resting membrane potential is always negative. How the resting membrane potential is generated? How they generated? Number 3 points there: contribution of the potassium diffusion potential, and contribution of the sodium diffusion through the membrane, and contribution of the sodium potassium pump. Please show the next slide. Yes, this one. Okay, keep this one. The only movement in case of contribution of the potassium diffusion potential, when you will say potassium diffusion potential, it is maintaining the resting membrane potential or originating the resting membrane potential, initiating the resting membrane potential. The only movement of the ions through the membrane is diffusion of the potassium ions. Only movement of the ions through the membrane is diffusion of the potassium ion. Because of the high ratio of the potassium ions inside to outside, that is 35:1. So the nearest potential corresponding to this ratio is minus 94 millivolts. So contribution of the potassium diffusion, contribution of the sodium diffusion through the membrane. The ratio of the sodium ions from the inside to outside the membrane is 0.1, and this gives a calculated Nernst potential for the inside of the membrane of about plus 61 millivolts. But the nearest potential for potassium diffusion of is minus 94 millivolts. In the normal nerve fibers, the permeability of the membrane to potassium is about 100 times as great as its permeability to the sodium. So using this value in Goldman equation gives a potential inside the membrane of 86 millivolts, which is near the potassium potential. Are you clear?
Yes,
Are you
sir.
clear?
Yes, sir.
Yes, sir.
Contribution of the sodium potassium pump. There is continuous pumping of the 3 sodium ions outside and 2 potassium inside, as I said previously. The fact that more sodium ions are being pumped to the outside than potassium inside causes continual loss of positive charges from the inside to the membrane, and this creates an additional degree of negativity, about minus 4 millivolts additional on the inside beyond that which can be accounted for by diffusion alone. So there is saying that Nernst potential. What is the Nernst potential? That is saying Nernst potential is the potential level across the membrane that exactly opposes the net diffusion of a particular ion through the membrane is called Nernst potential for that ion. The potential level across the membrane that exactly opposes the net diffusion. That means what diffusion diffusion concentration, your that will the Nernst potential will oppose that diffusion of a particular ion through the membrane is called Nernst potential. So Nernst potential for the inside the membrane and outside the membrane, it depends. It has different level. So action potential. The action potential is the rapid change in the membrane potential depolarization followed by repolarization. Action potential is rapid change in the membrane potential. Depolarization followed by repolarization. Please show the next slide. Next slide, please. Show the next slide. Show the next slide, please. Next slide. Next slide. Next slide, please. Yes, this one. Can you see the picture here? The typical action potential. Stages of typical
Yes,
action
sir.
potential.
Yes, sir.
Yes, sir.
You can see the below 90, below minus 90 millivolt, there is a resting membrane potential, isn't it?
Yes, sir.
And in case of 90, there is the initiation, that means threshold level. Minus 90 is the threshold level, and depolarization starts. Stage is okay. The depolarization starts, and it's getting higher, higher, and after this overshoot, it gets higher, it rises, and then repolarization occurs. Depolarization means the spike potential, and depolarization always starts from the resting stage. So there is three stages: resting stage, depolarization stage, and repolarization stage. As we know, the an action potential, this is very important. So concentrate on this. You must understand that an action potential is the rapid change in the membrane potential. Depolarization followed by depolarization. Do you know where the action potential occurs mainly? Where the action potential occurs? In the cells and throughout the body, whole body, every cell may have action potential. Like you have pain in the body. You have pain in the body. You have been bitten by a dog in any part of your body, and then action potential is increasing from that part of the body. Why this occurs? Because there is sodium leakage during injury. During injury, there is a sodium leakage from the cells occurs, and this sodium leakage causes depolarization of the cells. That means action potential initiates, and then after that repolarization occurs. That means potassium efflux, potassium leaks occurs, and then it stables. So that is the action potential. And how they generate, this is the it starts from the resting membrane potential. This is the resting membrane potential before the action potential begins. The membrane is said to be polarized during this stage because of the minus 90 millivolts negative membrane potential that is present in resting condition. And when the injury occurs or action potential begins, at this time depolarization stage, the membrane suddenly becomes very permeable to the sodium ions, very permeable to the sodium. Some sodium ions can't go out easily. But here, in case of injury or in case of depolarization starting or depolarization stage, at this time the membrane suddenly becomes very permeable to the sodium ions, allowing tremendous numbers of positively charged sodium ions to diffuse to the interior of the axons. Axons means the nerve axons. The normal polarized state of minus 90 millivolts is immediately neutralized by the inflowing positivity charge sodium ions. That means sodium influx occurs. Sodium influx occurs. In case of depolarization, sodium influx occurs. So what occurs? Can you understand? Do you understand? Am I clear up to this?
Yes, sir.
Someone say the depolarization stage, please. What may occur in the depolarization stage? So you are not understanding, and you are saying that yes, sir. Huh?
Sir, rapid influx of sodium ions through channel.
When it occurs?
Sir, the rapid rise of membrane potential in positive direction.
Yes, when you are injured or any of the stimulus causes this change in the membrane. This changes in the membrane, and this change in the membrane causes the huge level of potassium influx. That means in potassium getting inside the cell. So what it is here the sodium may go inside the axons of the nerve fibers so that you are feeling the pain. You can feel the pain. So signals going to your brain and you are feeling the pain. So up to the repolarization stage, you will feel the pain. So normal polarized state of the 9 is of minus 90 millivolts is immediately neutralized by inflowing positively charged sodium ions with a potential rising rapidly in the positive direction. This is called depolarization. Overshoot, what overshoot? In the large nerve fibers, the great excess of positive sodium ions. That means influx is occurring, so there is excess of positive sodium ions moving to the inside the cells, causing the membrane potential to actually overshoot beyond the zero level, that to become somewhat positive in some smaller fibers as well as in many central nervous system neurons. The potential merely approaches the zero level and doesn't overshoot to the positive state. Am I clear up to this? We will finish very soon.
Sir,
Yes.
what was the last line?
Last one was the potential in some smaller fibers, minute fibers, as well as in many central nervous system neurons, the potential merely approaches the zero level, and doesn't overshoot to the positive state. That means not gets at higher level. Okay, but most of them gets this overshoot stage. Overshoot stage, that means pain at its best. Okay, when sodium is influx occurs, you feel your pain in the nerve fibers, or you get your signals in nerve fiber, because action potential is initiated, and action potential goes up to this overshoot point. Some of the smaller fibers doesn't get this overshoot point, it gets stopped before that. The potential merely approaches the zero level and doesn't overshoot to the positive state. Am I clear?
Yes, sir.
Okay, repolarization stage. When a few thousands of a seconds, when a few 10,000s of a second after the membrane becomes highly permeable to the sodium ions, the channel begins to close. Sodium channel begins to close, and the potassium channel opens more than normal. Then rapid diffusion of the potassium ions to the exterior re-establishes the normal negative resting membrane potential. This is called repolarization of the membrane. That means the rapid diffusion of the potassium ions to the exterior of the cell re-established the normal negative resting membrane potential that was in the previous of the action before the action potential starting. So it again comes to the resting membrane potential after repolarization. Repolarization means potassium exflux, and depolarization means sodium influx. Depolarization means sodium influx. Repolarization means potassium efflux. Potassium efflux. Okay, efflux. Okay. Oh. So extra parts of action potential in special situation like spike potential. The sharp rise of the rapid fall of the action potential is known as sharp rise and rapid fall. When the sharp rise and the rapid fall occurs, this is called spike potential. Negative after potential, that means after depolarization, the rapid fall in repolarization is followed by a slow repolarization process. This is called negative after potential, that's after depolarization. And positive after potential, that means after hyperpolarization, is after reaching the previous resting level, it becomes more negative than resting level. This is called positive after potential, that is after hyperpolarization. You may be asked in the Bible, what is the cause of hyperpolarization? We'll read and discuss in the next class, inshallah, and we'll complete this one. We'll go through the muscle contraction process, actin fibers and how they act in actin fiber and myosin fibers, and then it will end our first chapter, that is introduction to the physiology. Then we'll go to the next chapter, blood, inshallah, next class. Okay.